Hello, I am Quim Martí, and today I'm going to talk about my bachelor thesis on whole genome doubling, the second most common event in cancer. My work is based on trying to model with mathematical tools this strange event that happens in cancer cells where the number of chromosomes suddenly duplicates. One of the most strange phenomena that arises in whole genome doubling is that cells tend to an optimal ploidy of 3.3. This means that on average, cells will have 3.3 copies of each gene. But why do cells do that? As some of you may know, cancer is a disease that is guided by the laws of evolution. Cancer cells are being constantly selected for higher resistance to the immune system, faster replication, better metabolism, and other important functions that are known as the hallmarks of cancer. These advantages are normally obtained by mutating specific genes. These are the oncogenes and the tumor suppressor genes. So, what is the advantage that whole genome doubling offers to the cancer cells? In theory, cancer cells that have their genome duplicated are selected due to the effect that whole genome doubling has on mitigating the Muller-Raychet. This is a process where asexual populations, like cancer, accumulate deleterious mutations due to a lack of recombination. Because there is no recombination, the progenitors of a cell with a deleterious mutation will have at least the same amount of mutations as its predecessors, if not more. Due to the deleterious mutations working in a recessive manner, they need to mutate all the copies of the same gene to produce a negative effect. This means that by duplicating the genome, the cell will have more copies of the same gene, thus reducing the odds of having all the copies of the same gene being mutated, and thus producing the negative effect. The main objective of my work is to model cancer evolution and whole genome doubling at the same time, taking into account how whole genome doubling affects the genome of the cell and its genes. This model could clarify if whole genome doubling really protects from the Muller-Raychet, or under which conditions is whole genome doubling selected, or which is the reason why whole genome doubling generates cells with a ploidy of 3.3. Thanks for watching this video, and if you have any question regarding the topic of cancer and whole genome doubling, don't hesitate to put them on the comment box. See ya!